This video tutorial is about translocation. Translocation occurs in the phloem and is the movement of assimilates throughout the plant. Assimilates are substances made by the plant using substances absorbed from the environment. These include sugars, mainly transported as sucrose, and amino acids. A part of the plant that loads assimilates into phloem sieve tubes is called a source. A part of the plant that removes assimilates from the phloem sieve tubes is called a sink. Sucrose is loaded into the sieve tube by an active process. This involves the use of energy from ATP in the companion cells. The energy is used to actively transport hydrogen ions out of the companion cells. This increases their concentration outside the cells and decreases their concentration inside the companion cells. As a result, a concentration gradient is created. The hydrogen ions diffuse back into the companion cells through special co-transporter proteins. These proteins only allow the movement of hydrogen ions into the cell if they are accompanied by sucrose molecules. This is known as co-transport. It is also called secondary active transport, as it results from the active transport of the hydrogen ions out of the cell and moves the sucrose against its concentration gradient. As the concentration of sucrose in the companion cell increases, it can diffuse through the plasmodesmata into the sieve tube. Movement of sucrose along the phloem is by mass flow. A solution of sucrose, amino acids, and other assimilates flows along the tube. The solution is called sap, and it can be made to flow either up or down the plant as required. The flow is caused by a difference in hydrostatic pressure between the two ends of the tube, which produces a pressure gradient. The water enters the tube at the source, increasing the pressure, and it leaves the tube at the sink, reducing the pressure. Therefore, the sap flows from the source to the sink. Sucrose entering the sieve tube element makes the water potential inside the sieve tube more negative or lower. As a result, water molecules move into the sieve tube element by osmosis from the surrounding tissues. This increases the hydrostatic pressure in the sieve tube at the source. A source is any part of the plant that loads sucrose into the sieve tube. In early spring, this could be the roots, where energy stored as starch is converted to sucrose and moved to other parts of the plant in order to enable growth in the spring. Perhaps the most obvious source is a leaf. Sugars made during photosynthesis are converted to sucrose and loaded into phloem sieve tubes. This occurs during late spring, summer, and early autumn, whilst the leaves are green. The sucrose is transported to other areas of the plant that may be growing, for example, the meristems, or to areas such as the roots for storage. A sink is anywhere that removes sucrose from the phloem sieve tubes. The sucrose can be used for respiration and growth in a meristem, or it can be converted to starch for storage in a root. Where sucrose is being used in the cells, it can diffuse out of the sieve tube via the plasmodesmata. It may also be removed by active transport. The removal of sucrose from the sap makes the water potential less negative or higher, so that water moves out of the sieve tube into the surrounding cells. This reduces the hydrostatic pressure in the phloem at the sink. Water entering the sieve tube at the source increases the hydrostatic pressure. Water leaving the sieve tube at the sink reduces the hydrostatic pressure. Therefore, a pressure gradient is set up along the sieve tube and the sap flows from higher pressure to lower pressure. This can be in either direction, depending upon where sucrose is being produced and where it is needed. It is even possible that sap flowing or could be flowing in opposite directions in different sieve tubes at the same time. Since the sap in one tube is all moving in the same direction, this is mass flow.